Hi, so my name is Aliah, and this summer I was evaluating Solanum sitiens introgression lines for frost tolerance. So tomato is very important agriculturally and economically, but the plants are highly vulnerable to low temperatures. So this leads to losses due to late frost and also restricts the growing season. So plant breeders, such as those in the Vogel lab, are trying to develop varieties that are frost resistant so they can overcome this challenge. So I screened 27 different introgression lines that each had a different chromosome segment from this wild species, Solanum sitiens, which is highly tolerant to drought, cold temperatures, and high salinity, which makes it of interest to breeders that are trying to get those traits into a cultivated tomato. And so we did a treatment at -3 degrees Celsius for 6 hours in a cold chamber. And then I rated the frost damage qualitatively on a scale from 0 to 5, as you can see here. I rated them an hour and then again 24 hours after the treatment to see how they were recovering. And then I also took tissue samples and measured the change in proline using an acid and anhydride colorimetric assay. And that was to see how, if there's a difference in how these plants were responding to stress, because proline is associated with like a stress response. So you can see there was kind of a difference in how the plants were responding to stress. And we were comparing everything to like our domestic parent. So our first one, LA4354, to see if there were any of these lines were significantly different from the domestic parent. And we did see some interesting results, but we were not able to find statistical significance for the frost damage ratings or for the proline assay, which kind of led us to believe that, or speculate that possibly frost tolerance in sitiens is not controlled by like a major effect QTL or just like one gene, that it may be several small effects genes that are distributed around the genome. So they might be distributed within these introgression lines. We might not be seeing one line that has that full expression of the trait, but it's still interesting. And this is still good information for breeders to know so they can look into this trait. So.
Yeah, sometimes science is like that.
Exactly.
You don't get the result you want or expect, but data is data. So yeah, so that's great. So you talked a little bit about some of the challenges that you had, like some things that didn't quite go what you thought. Was there anything that like went really well, or that if you were going to like replicate this or kind of redesign it, like what do you think like went really well about your project that you would want to continue doing?
I mean, I think we were definitely able to identify some lines that did worse than the parent. So that's good for like paring down the population for further screens. So you can get, kind of narrowing down on like which ones potentially are actually frost tolerant. And then I was pretty pleased with the results of the proline assay. We were able to see that our line of interest did have a higher increase in proline, even though it was not quite statistically significant. You can see here, this is our domestic parent and this is our line of interest having a higher increase. So I think there is kind of like a basis for what I was thinking, that like there is a difference in how these plants are responding to stress. Yeah.
Super interesting. Thank you so much. Also, your figures are beautiful. Very clear. Really like those a lot. Yeah. Yeah, they're really, really nice. Thank you so much for sharing with us. This is really, really great. Thanks.